بسم الله الرحمن الرحيم. دكتور أحمد النجار, Lecture of Histology and Cell Biology Department. This is the second lecture of the nervous tissue. Today we will discuss and explain nerve ganglia, neuroglia, and nerve degeneration. By the end of this lecture, the student will be able to 1. Define and describe the structure of the nerve ganglia. 2. Enumerate types of neuroglia. 3. Describe the structure and correlated function of astrocyte cells. 4. Define and describe the structure of blood-brain barrier. 5. Enumerate the steps and changes in nerve degeneration and regeneration. Nerve ganglia. Nerve ganglia is a collection of nerve cells and nerve fiber outside the CNS, covered by connective tissue sheaths. Nerve ganglia are two types: spinal ganglia and autonomic ganglia. Autonomic ganglia, sympathetic and parasympathetic ganglia. Again, nerve ganglia is collection of nerve cells and nerve fiber outside the CNS, covered by connective tissue sheaths. Two types of nerve ganglia: spinal ganglia and autonomic ganglia. Spinal ganglia and autonomic ganglia. Autonomic ganglia, sympathetic and parasympathetic. What is the difference between spinal ganglia and autonomic ganglia? 1. Site. Spinal ganglia represented by ganglia of cranial nerve 5, 7, 9, 10. Dorsal root spinal ganglia. Autonomic ganglia, sympathetic and parasympathetic ganglia. What are the functions of spinal and autonomic ganglia? Spinal is sensory function. Autonomic is motor function. Capsule of spinal ganglia is thick. Capsule of autonomic ganglia is thin. The nerve cells in spinal ganglia are pseudo-unipolar cells, but in autonomic ganglia, multipolar nerve cells. Nerve fiber in the spinal ganglia thick and myelinated, but in autonomic ganglia thin and non-myelinated. Synapse: spinal ganglia with no synapse, but autonomic ganglia there are synapses. Again, the difference between spinal ganglia and autonomic ganglia. Spinal ganglia represented by cranial nerve 7, 5, 7, 9, 10, and dorsal root ganglia. Autonomic, sympathetic and parasympathetic ganglia. Spinal ganglia is sensory function. Autonomic is motor function. Capsule of spinal ganglia is thick but in autonomic ganglia thin. The nerve cells of spinal ganglia pseudo-unipolar, but in autonomic ganglia multipolar. The nerve fiber of spinal ganglia thick and myelinated, in autonomic thin and non-myelinated. Synapse in spinal ganglia no synapse, and but in autonomic ganglia there are synapses. Synapse is a site of signal transmission from one neuron to another neuron or to muscle. Synapse can be classified according to site of synaptic contact, method of signal transmission, whether the synapse is excitatory or inhibitory. 1. According to site of synaptic contact can be classified into axodendritic synapse, axosomatic synapse, axonic synapse, dendrodendritic synapse. According to method of signal transmission, into chemical synapse, electrical synapse, mixed synapse. Chemical synapse, there is release of a neurotransmitter from presynaptic to postsynaptic cells to generate action potential. Neuron to neuron synapse or neuron to muscle synapse. This example. Electrical synapse, there is movement of ion from one neuron to another by gap junction. It is less numerous than chemical synapse but faster. Mixed synapse, electrochemical synapse. 3. According to whether the synapse is excitatory or inhibitory, depending on whether the neurotransmitter depolarized or hyperpolarized the postsynaptic membrane. Excitatory synapse is asymmetrical. Inhibitory synapse is symmetrical. Again, synapse, what is the definition of synapse? Synapse is the site of signal transmission from one neuron to another neuron or to the muscle. Classification of synapse according to site of synaptic contact into axodendritic synapse, axosomatic synapse, axonic synapse, dendrodendritic synapse. According to method of signal transmission into chemical synapse, electrical synapse, and mixed synapse. Chemical synapse, there is releases of neurotransmitter from presynaptic to postsynaptic cells to generate action potential. For example, neuron to neuron synapse or neuron to muscle synapse. Electrical synapse, there is movement of ion from one neuron to another by gap junction. It's less numerous than chemical synapse but faster than. Mixed synapse, electrochemical synapse. According to whether the synapse is excitatory or inhibitory, depending on whether the neurotransmitter depolarizes or hyperpolarizes the postsynaptic membrane. Excitatory synapse is asymmetrical. Inhibitory synapse is symmetrical. The structure of chemical synapse. Structure of chemical synapse. 1. Axon terminal. 2. Presynaptic membrane. 3. Synaptic cleft. 4. Postsynaptic membrane. Axon terminal as bulb-like swelling that occur either at the term end of the axon or long process. It contains many mitochondria and synaptic vesicles that contain the neurotransmitters. Again, axon terminal as bulb-like swelling that occur either at the term end or long process. It contain many mitochondria and synaptic vesicles, that is contain neurotransmitter. 2. Presynaptic membrane. It shows second density, actin-like filament. Presynaptic membrane, actin-like filaments. 3. Synaptic cleft. Synaptic cleft, small gap between presynaptic and postsynaptic membrane, 25 nanometer. Neurotransmitter diffuse across it, transfers by fine filament to guide the movement of neurotransmitter. 4. Postsynaptic membrane, second plasma lemma, actin-like filament and contain receptor for neurotransmitter. Again, what is the structure of chemical synapse? Chemical synapse consists of 4 structures: axon, terminal, presynaptic membrane, synaptic cleft, postsynaptic membrane. Axon terminal as bulb-like swelling that occur at the term end or long process. It contains many mitochondria and synaptic vesicles that contain neurotransmitter. Presynaptic membrane, second density, actin-like filament. 3. Synaptic cleft, small gap between pre and postsynaptic membrane, 25 nanometer. Neurotransmitter diffuse across it transfers by fine filament to guide the movement of neurotransmitter. Postsynaptic membrane, it shows second plasma lemma, actin-like filament and contain the receptors for neurotransmitter. Neuroglia. This picture showing some type of neuroglia around the neuron and around the blood vessels. Oligodendrocyte cells, microglia and astrocyte cells and ependymal cells. What is the definition of neuroglia? Neuroglia are the supporting cells of the nervous system. They are branching cells that bind the neuron together and to other elements as blood vessels. Again, neuroglia are the supporting cells of the nervous system. They are branching cells that bind the neuron together and other elements and to other elements as blood vessels. How to demonstrate it of neuroglia? By 2 techniques. 1. Immunohistochemical stain using antibodies against glial fibrillary acidic protein. 2 techniques is called gold or silver impregnation technique. The brown color is glial fibrillary acidic protein. Types of neuroglia. Neuroglia present in CNS and BNS. Types of neuroglia in CNS. 4 types. 1. astrocyte cells. 2. Microglial cells. 3. Oligodendrocyte cells. 4. Ependymal cells. Again, types of neuroglia in CNS: astrocyte cells, microglial cells, oligodendrocyte cells, and ependymal cells. Types of neuroglia outside CNS: 1. Schwann cells. 2. Satellite cells in the ganglia. 3. Pituitary cells in the posterior lobe of pituitary gland. Again, types of neuroglia outside CNS: Schwann cells, satellite cells, pituitary cells in posterior lobe of pituitary gland. Neuroglia types: Schwann cells and oligodendrocyte cells, myelin formation. Ependymal cells, CSF brain barrier. Astrocyte cells, blood-brain barrier. Microglia is phagocytosis. Again, Schwann cells and oligodendrocyte cells, myelin formation. Ependymal cells, CSF brain barrier. Astrocyte cells, blood-brain barrier. Microglia is phagocytosis. Myelin forming cells is Schwann cells and oligodendrocyte cells. Oligodendrocyte cells, most numerous glial cells. The difference between Schwann cells and oligodendrocyte cells: 1. Shape: Schwann cells is fillet cells. Oligodendrocyte cells is branched cells. Stellate shape with a small rounded nucleus formed from spongioblast cells. What is the site of Schwann cells and oligodendrocyte cells? Schwann cells present in peripheral nervous system. Oligodendrocyte cells present in CNS, central nervous system. Gray matter and white matter. Myelin: Schwann cells form myelin for one internodal segment, but oligodendrocyte cells form myelin for many internodal segment and for many axons. Neurolemma: Schwann cells, it forms neurolemma, but oligodendrocyte cell doesn't form neurolemma. Role of these cells in nerve regeneration: Schwann cells has very important role in nerve regeneration, but oligodendrocyte cells has no role in nerve regeneration. Again, myelin forming cells, Schwann cells and oligodendrocyte cells. What is the difference between Schwann cells and oligodendrocyte cells? Shape: Schwann cell is fillet cells. Oligodendrocyte cells is branched cells. Site: Schwann cells in peripheral nervous system. Oligodendrocyte cells in central nervous system. Gray matter and white matter. Myelin: Schwann cell can form myelin for one internodal segment, but oligodendrocyte cells, 4 myelin, 4 mini internodal segment, and 4 mini axon. Neurolemma Schwann cells, which 4 neurolemma, but oligodendrocyte cells doesn't 4 neurolemma. Role in nerve regeneration. Schwann cells has very important role in nerve regeneration, but oligodendrocyte cells has no role in nerve regeneration. Neuroglia, microglia origin blood monocytes, sites both gray and white matter, structure small spindle cells with central oval nuclei. Their processes are short and branching. Their processes arise from the tubule of the cells. Vital staining, trypan blue stain. What is the function of microglia? Phagocytosis. Again, microglia cells origin from blood monocyte, site in gray and white matter, structure small spindle cells with central oval nuclei. Their processes are short and branching. Their processes arise from the tubule of the cells. What is the vital staining? Trypan blue staining. What is the function of microglia? Phagocytosis. Ependymal cells, cubical epithelial cells, lining neural tube and ventricles. Ependymal cells is cubical epithelial cells lining neural tube and ventricle. They have basal process extending to gray matter. They have apical cilia to help in movement of CSF. Function: they assist information of CSF. Again, ependymal cells, cubical epithelial cells lining the neural tube and ventricles. They have basal process extending to gray matter, and they have apical cilia to help movement of CSF. What is the function of ependymal cells? They assist information of CSF. Satellite cells is small cells present in ganglia in BNS. They form thin layer around nerve cell body, capsule cells. What is the function of satellite cells? Protection, insulation, nutrition. Again, satellite cells is small cells present in ganglia in peripheral nervous system. They form thin layer around nerve cell body, capsule cells. What is the function of satellite cells? Protection, insulation, and nutrition. Astrocyte cells is the largest neurological cells. Astrocyte cells is the largest neurological cells. Astrocyte cells have two types. One, protoplasmic astrocyte cells in gray matter. Two, fibrous astrocyte cells in white matter. Again, astrocyte cells is the largest neurological cells. Two types of astrocyte cells: protoplasmic astrocyte cells in gray matter, fibrous astrocyte cells in white matter. What is the structure of astrocyte cells? Large and stellate in shape. Large and stellate in shape. Have cytoplasmic process with expanded tip. Perivascular foods. Have cytoplasmic process with expanded tip, called perivascular foods. Nucleus of astrocyte is large, rounded, central, and pale. The process envelope blood vessels, neuron, and synaptic area. Again, structure of astrocyte cells: large and stellate in shape. Have cytoplasmic process with expanded tips, called perivascular foods. Nucleus large, rounded, central, and pale. The process envelope blood vessels, neuron, and synaptic area. Structure of astrocyte cells: cytoplasm contains intermediate filaments composed of glial fibrillary actin protein GFAP. The brown color in this picture is GFAP. Again, cytoplasm contain intermediate filament composed of GFAP. This brown color in this picture is called GFAP. Function of astrocyte cells: they are supportive for nervous tissue. 2. They serve as insulators for synaptic contact. 3. They have nutritive function. 4. They play role in scar formation in central nervous system. This process is called gliosis. 5. Maintain the blood-brain barrier. Again, what is the function of astrocyte cells? 1. Support of nervous tissue. 2. Insulator for synaptic contact. 3. Nutrition function. 4. Play role in scar formation in CNS. This process is called gliosis. 5. Maintain the blood-brain barrier. This table showing the difference between types of neuroglia. 1. Astrocyte cells, microglial cells, oligodendrocyte cells, ependymal cells. Shape: astrocyte cells, large stellate in shape. Multiple processes end by foot-like expansion on the blood vessels. Microglia, small and oval in shape, processes arising from the two pools, the cell body and the processes have minute spine. Oligodendrocyte cells, small cells, few short processes. They are aligned in row between the axon and the white matter. Ependymal cells, epithelial-like cuboidal cells, epithelial-like cuboidal cells lining the brain ventricle and the central canal of spinal cord. Apically have microvilli and few cilia, while basal have numerous enfolding without a basement membrane. What is the function of astrocyte cells? Supportive, nutritive, metabolic, formation of blood-brain barrier. Microglia is phagocytosis of bacteria, apoptotic and malignant cells. Oligodendrocyte cells formation the myelin sheath in the white matter of CNS. Function of ependymal cells formation of cerebrospinal fluid. Again, this table showing the difference between neuroglial cells, astrocyte, microglia, oligodendrocyte cells, ependymal cells. Shape: the astrocyte cells large stellate, multiple process ended by food-like expansion around the blood vessels. Microglia oval, small processes arising from the two pole, cell body and processes have minute spine. Oligodendrocyte cells, small cells, few short processes. They are aligned in row between the axon and in the white matter. Ependymal cells as epithelial-like cuboidal cells lining the brain ventricle and the central canal of the spinal cord, have microvilli and few cilia, while basal have numerous enfolding without a basement membrane. What is the function of astrocyte cells? Supportive cells, nutritive cells, metabolic cells, and formation of blood-brain barrier. Function of microglia, phagocytosis of bacteria, apoptotic cells, and malignant cells. Function of oligodendrocyte cells, formation of myelin sheath and white matter of CNS. Function of ependymal cells, formation of cerebrospinal fluids. Blood-brain barrier. Blood-brain barrier is a selective barrier between the blood and the brain. That is the function of blood-brain barrier. Prevent passage of toxic substance from blood to the brain. Again, blood-brain barrier is a selective barrier between blood and brain. What is the function of blood-brain barrier? Prevent passage of toxic substance from blood to the brain. What is the structure of blood-brain barrier? Structure of blood-brain barrier consists of three parts. 1. Endothelial cells of the capillary joined by zonular occlusions. 2. Complete basement membrane of endothelial cells. 3. Feet of astrocyte cells that form complete envelope around the capillaries. Again, structure of blood-brain barrier. 1. endothelial cells of the capillaries joined together by zonular occludens. 2. Complete basement membrane of endothelial cells. 3. Feet of astrocyte cells. That is 4. Complete envelope around the capillaries. This picture showing blood-brain barrier. Endothelial cells, complete basement membrane of endothelial cells, feed process of astrocyte cells that is form envelope around blood capillaries. Degeneration and regeneration of the nerve tissue. Any damage in the nerve of the central nervous system is permanent. Any injury in the peripheral nerve is followed by regeneration. Again, any damage in the nerve of the CNS is permanent. Any injury in the peripheral nerve is followed by regeneration. Cut of a peripheral nerve will induce any cut of a peripheral nerve will induce three types of degeneration. Traumatic degeneration at the site of injury, retrograde degeneration in the cell body or proximal part to cell body. 3. Wallerian degeneration in the distal segment of the cut nerve. Again, any cut of a peripheral nerve will induce traumatic degeneration, retrograde degeneration, Wallerian degeneration. Traumatic degeneration occurs at the site of injury. Retrograde degeneration in the cell body and proximal segment of cut nerve. Wallerian degeneration in the distal segment of the cut nerve. What is the definition of retrograde degeneration? Changes which occur in the cell body after injury of its axon. How occur? The cells swell, withdraw its process and become spherical. Nucleus become eccentric and bikinotic. Nasal granules break down into fine granules and disappear. Chromatolysis. Golgi complex disintegrates and finally disappears. Neurofibrils disintegrate and finally disappear. Finally, the cell body degenerates and its axon undergo Wallerian degeneration. Again, retrograde degeneration. What is the retrograde degeneration? Retrograde degeneration, this is the changes which occur in the cell body after injury of its axon. The cells swell, withdraw its processes and become sphericals. Nucleus become eccentric and bikinotic. Nasal granules break down into fine granules and finally disappear. Golgi complex disintegrate and finally disappear. Neurofibrils disintegrate and finally disappear. Finally, the cell body degenerate and its axon undergo Wallerian degeneration. This picture showing retrograde degeneration, that is the occur in axon proximal to cell body. Wallerian degeneration. What is the Wallerian degeneration? Wallerian degeneration is the degenerative changes that occur in the distal segment of the nerve fiber. It includes the following: axon, myelin sheath, and Schwann cells. Axon undergoes rapid degeneration in few days. Myelin sheath, it is fragmented into fat droplets. The degenerating products of both axon and myelin sheath are removed by macrophage cells. Again, Wallerian degeneration is the degenerative changes that occur in the distal segment of cut nerve fiber. It includes the following: axon undergo rapid degeneration in few days. Myelin sheath, it is fragmented into fat droplets. The degeneration products of both axon and the myelin sheath are removed by macrophage cells. Schwann cells and neurolimmal sheath don't degenerate. It proliferates to form a thick tube distal to the injury to the effector cells. This tube acts as a guide for the regenerating axon. This axon will spread for the proximal stump of the cut nerve. Again, Schwann cells and neurolimmal sheath don't degenerate in Wallerian degeneration. It proliferates to form a thick tube distal to the injury to the effector cells. This tube acts as a guide for the regenerating axon. The axon will spread, forming the proximal stump of cut nerve. This picture showing Wallerian degeneration occur. Regeneration. Factor affecting regeneration. 1. The presence of the neurolimmal sheath and Schwann cells. 2. The nerve fiber is partially cut. 3. The small distance between the two cut ends. 4. There is no infection because or as infection lead to the formation of fibrous tissue which fills the gap between the two ends. Again, factors affecting the regeneration. 1. The presence of neurolimmal sheath and Schwann cells. 2. The nerve fiber is partially cut. 3. Small distance between the two cut ends. There is no infection, as infection lead to formation of fibrous tissue which fills the gap between the two ends. What is the mechanism of regeneration? 1. The neurolemma proliferate, forming the guide regenerative tubes. The nerve cells resumes its normal structure. Several branches, axon, sprouts grow at the rate of 1.5 mm in days. 4. Several axon sprouts may enter the tube. Only the proper axon will reach the proper nerve ending. 5. Schwann cells then form myelin sheaths around the formed axon. Again, what is the mechanism of regeneration? 1. The neurolemma proliferates, forming the guide regenerative tube. The nerve cells resumes its normal structure. Several branches grow at the rate of 1.5 mm in day. Several axon sprouts may enter the tube. Only the proper axon will reach the proper nerve ending. Schwann cells then form myelin sheaths around the formed axon. This picture showing degeneration occur in the axon of the nerve cells and regeneration occur after cutting of the nerve cells. Thank you.